research thesis project today. And Petra started working in our lab about 2 years ago in January 2023, so a little over 2 years ago. And as you'll see, she's completed quite a bit of work during those 2 years, which included 2 full-time stints during the summer, which were funded by competitive fellowships. So she was awarded a fellowship from UVM Cancer Center in summer 2023, and I think she was the only undergrad to receive that award at that point. And then she was awarded a summer fellowship from Microbiology and Molecular Genetics, the Nicole Ferland Award in summer 2024, which is a very competitive award. Her project has really focused on testing mechanisms of resistance in cancer cells to a new therapeutic drug that the lab discovered several years ago. And she's worked closely with Kira Fisher on that project. And then outside of the lab, Petra also stays really busy. So she's the captain of the women's club lacrosse team, who won the national championship last year. She's also been involved in the Women in STEM club here at UVM. So the way the format is going to work, Petra's going to give a presentation, and then we'll open it up for questions from the general audience. And then once she finishes answering those questions, then we'll have her meet just with her committee to answer questions from the committee members. And then after that, hopefully everything will go great, and she will have completed her distinguished undergrad research requirement. So Petra, I'll turn it over to you.
It's really nice to meet you all. And thank you all for coming to my presentation. I'm excited to give you a little insight to what I've been working on for the past 2 years in the lab. And like Jason mentioned, the work that I've been doing has accumulated into a project that I've titled Understanding the Mechanisms that Can Lead to Main Resistance in Chromosomal Unstable Cancer Cells. Then I want to first start off pretty general with talking about mitosis, which is one of the processes cells use to grow and divide. And this starts with the cells replicating their genetic information and then aligning them in what's called a metaphase plate in the center of the spindle, so that it allows for the equal separation of the genetic information to form 2 genetically identical daughter cells. And this process is highly regulated, and when there's errors, it can lead to uncontrolled cell division, or what we know as cancer. And mitosis relies on the formation of a mitotic spindle, and here are some components of a mitotic spindle. In dark blue, you can see the chromosomes, which are the genetic information, or condensed DNA, and that's attached to a group of proteins called the kinetic core, and the kinetic core is attached to the microtubules, which makes up really the structure of this spindle. So as I mentioned, these compose the spindle, and microtubules themselves are composed of tubulin protein subunits. So 2 tubulin proteins are together, and these pairs are added to the microtubules for the growth, and then taken away in depolymerization. There's the plus end of microtubules that have growth and shortening, and then the minus end is just removal of these protein subunits, and this leads to kind of periods of random growth and shrinking, which is termed dynamic instability. And this is how chromosomes move within the spindle to ensure for proper cell division. Since this is so important, the spindle is highly regulated, and when there are errors with the movement of chromosomes, it can lead to missegregation of chromosomes and genetically unidentical daughter cells. So typically, chromosomes are separated so that the 2 daughter cells have the same number of chromosomes, but when there's issues with this process, some daughter cells will have more chromosomes than they typically have, while others will have less. And this is termed aneuploidy. And when there's a frequent loss of gain of chromosomes. It's termed chromosomal instability. And this is a frequent hallmark of cancer cells and proves to be a promising target to treat cancer cells while bypassing those healthy euploid cells that have the correct number of chromosomes. And it's also important in tumorigenesis. So here we have a stable tumor cell, and then through periods of division, it might acquire sin, in which it allows the loss and gain of chromosomes lead to a heterogeneous tumor that leads to aggressive tumor behaviors like not being responsive to drug or metastasis and other things that are really difficult to treat for patients who have these cancers. And like I said, the difference between cancer cells that have the loss or gain of chromosomes as opposed to our normal cells that have the same number of chromosomes proves to be a potential target that we can use in the clinic. And when we focus on the spindle formation of sin cells, where we're trying to look for proteins that are more important in the sin cells as opposed to the normal cells, it might not be as important for that spindle formation, therefore bypassing the healthy cells and hopefully killing the cancer cells. So based on that, our lab previously looked at some specific proteins that they thought to play a role in the formation of the spindles in the sin cells. By knocking those specific proteins down, they looked at a group of proteins called kinesins, which are important in mitosis, and they found that KIF18A, when they inhibited the function of that protein, it decreased the growth rate of a sin cell line, the MDA-MB231 cells, and these are triple-negative breast cancer cell lines. While that was happening, another group also looked at, did a database screen to look at 100 different cancer cell lines and compared the essential genes for the aneuploid cells, the cells with differing chromosome numbers, as opposed to the healthy euploid cells, and found that, again, KIF18A was more essential for the growth and division of aneuploid cells as opposed to euploid cells. So what is KIF18A? KIF18A is part of the kinesin 8 motor protein family. It walks across the microtubules by hydrolyzing ATP, and it localizes to that plus end that I was talking about earlier, and other microtubules that also attach to the kinetochores and the chromosomes. And as you can see in the immunofluorescence images here, KIF18A is labeled by that bright puncta seen in the middle, and then the tubulin or microtubules in the middle panel, and then finally is this overlay where you can see the green KIF18A on the edge of the microtubules in the middle. And this protein regulates the microtubule dynamics and specifically suppresses that growth that happens. And this helps the chromosomes move towards the spindle equator during mitosis. After knowing this, our lab decided to inhibit the function of KIF18A and look for the phenotypes that we see in normal cells as opposed to the phenotypes that result in the KIF18A knockdown cancer cells. And we found that when we inhibited KIF18A in the normal cells, there were minor spindle defects and didn't decrease rates of proliferation very drastically in normal cells, as opposed to when we knocked down KIF18A in sin cells, we saw major spindle defects, which resulted in centrosome fragmentation. So instead of seeing those two poles, we saw more than two poles, and then micronuclei, which resulted in mitotic delay, and then eventually mitotic arrest. And these cells did not continue to divide, and therefore the proliferation rates decreased. And this can be seen in this video taken by a member in our lab, where a compound was introduced to cells inhibiting KIF18A, and you can see some of these cells are having trouble aligning the chromosomes, and some two of these cells don't even divide in the video because they're not able to pass that checkpoint. Green is DNA, and then this magenta is reticulin. After these findings came out, it showed to be a promising therapeutic target. And because of that, some companies have created a compound called sovalnecib, or compound 14, and I'll be using those terms interchangeably throughout the rest of the presentation. And multiple drugs are already in clinical trials, which is exciting, and based on modeling, it has been hypothesized that sovalnecib binds between the alpha-4 and the alpha-6 region of the microtubule binding region of the motor domain of this kinesin protein. So essentially locks the kinesin onto the microtubules, and due to the flux of the microtubules, the kinesin ends up relocalizing to the poles of the spindle. And this can be seen in this video taken by other members in our lab, and this green that you'll see starts in the middle and is the KIF18A, and then it moves to the sides with an addition of compound. This is a very exciting finding. However, cells have been shown to gain resistance to this compound. And in order for it to be effective in clinic, we want to make sure we understand the mechanisms of resistance and hopefully co-target multiple pathways and make this drug more effective for patients. So my project really has been exploring how these cells could potentially become resistant to the drug. And over the past 2 years, I've looked at 3 potential hypotheses that have come out of our lab. The first has been through the potentially a compensation mechanism with a similar kinesin protein. The second is based on the upregulation of a specific gene in the resistant cells and understanding what role that plays in resistance. And then finally, understanding how changes in the microtubule polymerization rates can confer to resistance. And then the first one I'm going to talk about is this closely related mitotic kinesin hypothesis. And the first step was to develop the resistant lines that we could use in further experiments. So again, the cells that I used particularly were the MDA MD231 cells, which were the triple-negative breast cancer cell lines. And we plated that with media and then treated that with sovalnecib, or compound 14, inhibiting the KIF18A. And then the cells that survived over time were selected to create eventually resistant line that could continue to divide in compound. And we also wanted to check that the drug, that resistant cells still had their KIF18A inhibited by the compound, but were able to divide. So that would suggest they had compensatory mechanisms of resistance, as opposed to just having a mutation in KIF18A that allowed the drug not to bind. And we wanted to also check that these cells showed a phenotype similar to the normal cells when treated with compound, and this was done by Kira. And we did this by doing a mitotic index assay, which I will continue to use throughout the presentation. What a mitotic index assay is we seed the cells, and then 24 hours later, we will treat them with compound or other drugs, and then fix them onto cover slides, or essentially freeze them onto a cover slide, and then tag them with primary and secondary antibodies to tag proteins to look at the spindle formation of these cells. So I'm able to count all the cells seen in a field of view, and then count the specific cells in mitosis to determine the rate of mitotic arrest in each population. And so what we saw with when cells aren't treated with any drug or compound, they should not have high rates of mitotic arrest. However, the sensitive cells, when we treat with the KIF18A inhibition, we see like about 15% mitotic arrest. However, when we do that with the resistant cells, there's a rescue of the phenotype, and they're able to continue through mitosis and not get arrested or stuck at checkpoints. Additionally, we can count for those cells that have more than 2 spindles, and that is the percent seen to have multipolar. And that's based on, out of the cells in mitosis, which cells have more than 2 poles. And again, resistant cells had decreased levels of that too, which means this cell line can continue to divide normally in the presence of the drug. So how are they able to do that? And again, first we looked at hypothesis that potentially a similar kinesin is being compensated for that loss of the kinase, or the KIF18A. So KIF18B is the protein we came up with, and this is also within the same kinesin family that KIF18A is a part of, and has been shown to have a lot of similar commonalities in the binding and the motor regions of this protein, and you can see this in the sequence alignment here, where the red indicates the amino acids that are conserved between the 2 proteins. And the difference between this protein and KIF18A is that KIF18B binds to, instead of the kinetophore microtubules, it regulates and localizes to the plus end of the astral microtubules, which are the microtubules that kind of are on the outside, the opposite facing. And you can see that in this immunofluorescence image, where this green puncta is kind of facing this way and outside of the poles, as opposed to where KIF18A is where the DNA, or that blue region, is typically. It is important to note that we do know the compound does not inhibit KIF18B, or does not impact KIF18B. Studies have found that this specific amino acid residue, the mutation changes, and even that switch allows for the compound not to bind, and therefore the compound does not impact KIF18B. So based on this, due to the similar structure and function, we hypothesized that resistant cells are using KIF18B in place of KIF18A when KIF18A is knocked down to regulate those microtubules and continue to divide normally. And based on that, we would expect that in resistant cells, if we treat it to compound, effectively knocking down KIF18A and knock down KIF18B, we should see the similar phenotypes with increased mitotic arrest. And again, I did this by doing a mitotic index assay, and here's a little bit more of a view, which is a little bit challenging to see with the lighting. But again, I had the MDA231 cells, seeded them. After 24 hours, I treated them with compound control drug, a control siRNA, and siRNA targeting KIF18B, which inhibits the function of that protein. And then after 24 hours, I fixed those cells, treated them with primary and secondary antibodies to image the proteins of interest within the cell, and then again counted the cells seen in mitosis. And we did this, and at first we want to make sure that the siRNA that we're using to target KIF18B was in fact targeting the protein that we wanted to see inhibited, and Kira quantified this by looking at those immunofluorescence images that we had, and finding the signal intensity of KIF18B within the cell and subtracting it from the signal in the background to determine the changes in protein levels within the cell. And we found that with 24-hour knockdown of using our siRNA and against KIF18B, it did effectively decrease the amount of KIF18B within the cell. And that just further supports the finding that we found in the mitotic index assay, where the resistant cells, when knocked down with KIF18B, did not show any change in mitotic arrest, which did not support our hypothesis that this protein was important for resistance. if our hypothesis was correct and that resistant cells were using KIF18B, we would see an increase in mitotic arrest similar to the one seen here in the resistant cells and compound. But as you can see, that was not found. And we also quantified the percent multipolar. So the mitotic index assay did not support our hypothesis, but the increase in cells seen with multipolarity was higher, which could suggest that KIF18B plays some role in centrosome fragmentation and control of that. However, it is important to note that with such low levels of cells seen in mitosis with these images, even one or two cells with multipolar spindles kind of swayed the data, so more replicates would have to be done to confirm that finding. And then we wanted to explore our other part of our hypothesis. Does KIF18B relocalize to the area where KIF18A is seen normally? And by staining these cells with a kinetochore protein and staining for KIF18B, we were able to find that the distance between KIF18B and the kinetochore protein SIMC decreased in resistant cells, which does support our part of the hypothesis that KIF18B relocalizes to the area where KIF18A typically is. And you can see this in the immunofluorescence images. The top two rows are sensitive cells, and the magenta KIF18B is on the outside of the cells, whereas this bottom row are the resistant cells in compound, and you can see the purple is not only on the outside but also right up close to that green fluorescence, which is the kinetochore proteins. So this supports the idea that KIF18B does relocalize to resistant cells. So what does this all mean? We found that KIF18B does not contribute to the mitotic arrest phenotype in resistant cells, but could potentially play a role in multipolarity seen. And KIF18B does relocalize to where KIF18A typically is, closer to the kinetochores. So this suggests that KIF18B might play a small role in resistance mechanisms, but is not the primary cause of resistance. So based on this relatively null finding, we decided to look at a different mechanism, and this was based on RNA-seq analysis done, where we looked at the two different cell lines. We looked at the MDA and B231s, which I use, and then a colorectal cancer cell line titled HT29 cells, and we compared the we looked at the increase in certain genes with resistant cells as opposed to sensitive cells, and this would show us potentially that certain genes that are upregulated resistant cells could potentially be more important as opposed to the sensitive cells, and potentially conferring resistance mechanisms. And with these two cell lines, we saw one specific gene upregulated in both of them, and this was called MAGE-A3. And the research shows that MAGE-A3 is known to form a ubiquitin ligase complex with other MAGE proteins and other proteins to tag P53 with ubiquitin, which leads to the degradation of this protein. And P53 is an important protein because this is a tumor suppressor protein, which essentially monitors the cell during mitosis for any DNA damage. And if there are DNA damage, this protein should trigger programmed cell death, and so that no cells are continued to divide with damaged DNA. And this protein is seen to be mutated in stem cells, and actually KIF18A dependency correlates with this mutation, which makes this hypothesis very interesting. And if MAGE-A3 inhibits P53, these cells should continue to divide. And based on this understanding, we hypothesized that resistant cells might rely on MAGE-A3 to continue to divide when in the compound sogomesiv. So if KIF18A is inhibited in these cells, we believe that they were upregulating MAGE-A3, which in return decreased p53 levels, which allowed the cells to continue to divide. And based on that, we thought that we should knock down KIF18A and MAGE-A3, in which case p53 levels should increase because there's no MAGE-A3 inhibiting it, and thus increase the mitotic arrest seen in resistant cells. And using same mitotic index assay, we looked at this data, and first we wanted to again confirm that siRNA that we were using to target MAGE-A3 was working, and we did the same quantification with the signal analysis and found a significant difference between control siRNA and MAGE-A3 siRNA at 24 and 48 hours. However, there is such a great overlap and not a drastic decrease in overall average signal intensity that it's hard to determine if MAGE-A3 was knocked down enough to have any impact. And this also could be skewed based on the antibody we used. Our antibody might be not just tagging MAGE-A3 but other proteins, so it might not be so specific. So it's hard to determine, again, if MAGE-A3 is knocked down. So we wanted to look at a qPCR analysis to see, really at the molecular level, are our siRNAs targeting what we wanted to. And based on an untreated control, the control siRNA was knocking down the MAGE-A3 by 40%, and the MAGE-A3 specific siRNA was knocking it down by 80%, which makes this data hard to interpret because the control really shouldn't be knocking down MAGE-A3 transcripts at all. However, the MAGE-A3 siRNA did knock down 80%, so we did want to see if we treated cells with compound and MAGE-A3 siRNA did impact mitotic arrest. And we found that resistant cells still did not have any change in mitotic arrest when treated with compound and MAGE-A3, which does not support our hypothesis that is important to resistance. Again, we wanted to just double check that is there still functional MAGE-A3 within the cell that's helping it to divide. And so I did another signal analysis of this based on the photos that we took in this assay, and got some interesting results. And we found that there's a significant difference between the sensitive cells with the MAGE-A3 siRNA, but not with the resistant cells, which should not be the case, and further supports the idea that the knockdown cannot be confirmed. Therefore, the conclusion based on the mitotic arrest data can also not be confirmed. So again, MAGE-A3 does not appear to have a mitotic or contribute to mitotic arrest phenotype, but due to the knockdown confirmation, we cannot say that this is true yet. And it might play some role, but as far as we know, it probably isn't the primary role of resistance. So we move on to our third and final hypothesis. And this is based on another difference between sensitive and resistant stem cells. One thing about stem cells is that a 2014 study found it had increased rates of microtubule polymerization as opposed to non-stem cells. And this makes them susceptible to the loss of KIF18A because KIF18A controls microtubule polymerization. And this can be seen in these images or these videos where the top one, there's no KIF18A added, and you can see this random growth and shrinking is happening, whereas opposed to when we have these, we add the fluorescent KIF18A in green, there isn't this random growth and shrinking of microtubules. So we believed that KIF18A is suppressing the stem cell microtubules, and if we eliminated KIF18A, it's further increasing their already fast microtubule polymerization growth. And somebody in the lab previously has already looked at this data by tracking some comments at the end of microtubules. It's kind of challenging to see, but the movement of those was tracked and found that, in fact, sensitive cells, when treated with KIF18A, further have even increased rates of microtubule polymerization when KIF18A is knocked down, and the same effect with the compound as well. So this leads us to the hypothesis that potentially resistant cells are reducing their microtubule polymerization rates, which makes them less sensitive to the loss of KIF18A, which is that microtubule suppressive protein. So to really understand this further, we want to look at sensitive cells when exposed to a microtubule suppressing drug, Taxol. Will it counteract the effect of that KIF18A inhibition and decrease their sensitivity to the drug, and therefore decrease mitotic arrest? So we already know that when we treat the cells with the silver-nested compound, they have an increase in microtubule polymerization, but if we decrease them, will we see a decrease in mitotic arrest? And to do this, I first did a proliferation assay where I seeded cells into wells, and 24 hours later added compound and or Taxol, and then put them into a biosol so that they could be imaged for every 2 hours for a period of 96 hours. And the microscope has a software attached where, based on the contrast of a bright field, they're able to count the cells at every time point so that you're able to measure the growth rate of these cells. And first I just looked at sensitive cells treated with Taxol to determine what dosage really impacted the growth of the sensitive cells, and we saw a significant decrease in proliferation between the addition of 2 nanomolars of Taxol and 6 nanomolars of Taxol, so very low doses. And based on that, we wanted to use concentration right in the middle, so we used 4 nanomolar of Taxol to treat the sensitive cells with compound to see if it changed the rate of mitotic arrest. And we found that, again, doing mitotic index assays, when we treated sensitive cells with the compound and the Taxol, it decreased the rate of mitotic arrest, which does support our hypothesis that slowing the microtubule polymerization helps these cells divide and be less sensitive to KIF18A. We also did this with resistant cells and saw actually a significant increase in mitotic arrest with compound and Taxol, although it's very, very small. That could suggest potentially that resistant cells already have the reduced microtubule polymerization rates, and then by adding Taxol, further reducing could cause more issues in division. However, mitotic arrest doesn't always correlate with growth rate changes. It could, or it could just mean that the Taxol is arresting the cells in a different part of the cell cycle where I can't see on the images. The formation of the spindle might have not happened already, in which case we want to look at the proliferation assays to see if the addition of Taxol did in fact help these cells divide. And the first replicate I did was on the left here, and where I had the sensitive cells, treated them with a control, treated them just with compound, and then varied increasing doses of Taxol. And you can see that with compound, it drastically decreased their growth rate, and then with the addition of 2 nanomolar Taxol, it increased that proliferation just slightly, but that does correlate with what we found in mitotic index assays. However, after 2 more replicates of this experiment, that pattern was difficult to decipher. However, this data is slightly concerning for 2 reasons. One is that there was no statistical significance between the cells treated without any compound as opposed to the cells treated with compound, which does not mimic any of the other data that we found for these cells. These cells are supposed to be sensitive to compounds, and we don't see that. And the other kind of red flag is that there's variation between experiments. So you can kind of see the clustering of data between experiments, which makes this result a little bit harder to confirm. Another reason why this doesn't feel like it follows what we expect is that when Kira did this same exact experiment with HG29 cells or colorectal cancer cells, we did see what we would expect based on our hypothesis. She saw a drastic decrease when treating these cells with compounds, and then at addition of 2 nanomolar and 4 nanomolar Taxol, there was this rescue and continued growth rate. It is important to note that HG29 cells have been seen to be more sensitive to the compound regardless, so it might just be easier to see this trend based on the difference in the cell type, but this would support our hypothesis that a decreased microtubule polymerization rate confers resistance. And here's also some images of what's taken in the microscope and how cells are counted and when it was counted at the time. And so to conclude, there is evidence that low doses of Taxol does rescue the mitotic arrest seen in sensitive cells, but we don't know for sure if maybe this is just arresting at different times. Due to variability within the data that I found, it cannot yet be confirmed. So in conclusion, I looked at 3 different hypotheses for mechanisms of resistance that these cells are using. The first I looked at was HIF-18B, and we thought that potentially resistance is conferred through the compensation of this, and the data does not show that it contributes to mitotic arrest, but potentially plays a role in the multipolarity seen. And then we looked at the upregulation of MAGE3, which really controls the P53 levels. And we also found that MAGE3 does not appear to contribute to mitotic arrest. But again, that knockdown was hard to confirm. And then finally, the most promising of these hypotheses was if resistance was correlated to the changes in microtubule polymerization rates. And it does appear that low doses of Taxol partially rescues that phenotype, and that decrease in microtubule polymerization rates can decrease the sensitivities. Cells, if I were to continue with this hypothesis, I would do more replicates of the assay that I did to reduce that variation and really confirm the findings that I had. I'd also want to look at the resistant cells and their response to Taxol alone based on that change in mitotic arrest. We want to see if resistant cells had different effects with very different concentrations of Taxol. And then finally, I would also treat cells with a microtubule destabilizing drug, nicotazole. So nicotazole kind of does the opposite of Taxol. And if resistant cells have decreased rates of microtubule polymerization, we make them sensitive to the compound by treating them with nicotazole, which increases the rates of microtubule polymerization. And that concludes my presentation. I want to thank the STEM class so much for an awesome 2 years. We got to learn from such great scientists, and then my committee members for helping me through this process, and then my friends and family for always being supportive through